So students, in the previous chapter, which is called as Principles of Inheritance, so in that chapter you have already come across the word called inheritance. So you have learned so many aspects about the genetics in the chapter of Principles of Inheritance, and the chapter that is the Principles of Inheritance mainly talks about the classical genetics, the traditional genetics. You learnt about the various experiments conducted by many different scientists, right? It can be Morgan, it can be Mendel, right? Many other scientists also you have come across, and you also noticed what all different types of experiments they conducted with regards to genetics. See, Mendel, he was very much sure about the inheritance. So I need not define what is inheritance again. You already know what is inheritance. Inheritance is the process by which the parents pass on the characteristics from their generation to the next generation. Right? Parents generation in the offspring galige, progenige, characteristics transfer agodake navu the process that is called as inheritance, right? So while talking about this inheritance, Mendel irbodu, Morgan irbodu, right? All these people, they talked so much about inheritance. They gave so much idea about the different patterns of inheritance, right? And even they told about the interactions of the alleles and all, but they did not clearly know what exactly is this factor that is responsible for the inheritance. That means, what is the molecular structure, what is the molecular nature or characteristic of this particular factor which was later called as gene. ಅವರು ಫ್ಯಾಕ್ಟರ್ಸ್ ಅಂತ ಕನ್ಸಿಡರ್ ಮಾಡ್ತಾ ಇದ್ರು, ಅದಕ್ಕೆ ಜೀನ್ ಅಂತ ಮತ್ತೆ ಇಟ್ ವಾಸ್ ಲೇಟರ್ ಕಾಲ್ಡ್ ಆಸ್ ಜೀನ್, ರೈಟ್? ಅಂಡ್ slowly, after many years, it was known to be what gene is nothing but DNA, right? In this chapter, in detail we are studying about the molecular structure of that factor that is responsible for inheritance. So which factor is that? See, you already know the two main factors or two main types of molecules which can be the genetic molecules. They are nothing but DNA and RNA. Predominantly, DNA is the most efficient genetic material in most of the living organisms in the living world. Though RNA is found to be a very rare molecule which can be considered to be the genetic molecule, but still it is there in some of the organisms such as viruses, and those viruses which have got RNA as their genetic material, and those viruses are called as retroviruses. The viruses with the genetic material RNA are called as retroviruses, and these are the only group of organisms which have got RNA as the genetic material, and all other organisms, they have got DNA as the genetic material, right? So we will understand why this DNA is so dominant over the RNA as genetic material in this chapter. We will understand the detailed chemical structure of DNA. What is it made up of? What kinds of chemical bonds are there in DNA and RNA? And other characteristic features of DNA and RNA, that is the expressibility of DNA and RNA. Expressibility means what? See, DNA is often also called as the master molecule in the body of living organism. Idikke master molecule anta yaake kariteve anta helidre, it is DNA which completely and solely is responsible for controlling each and every activity, each and every characteristic of any living organism for that matter. So ಅಷ್ಟೊಂದು ಇನ್ಫಾರ್ಮೇಶನ್ ಅದು ಹೋಲ್ಡ್ ಮಾಡ್ತಿರ್ಬೇಕಾದ್ರೆ ಇಟ್ ಮಸ್ಟ್ ಬಿ ಹ್ಯಾವಿಂಗ್ ಸಮ್ ಕೋಡೆಡ್ ಇನ್ಫಾರ್ಮೇಶನ್, ಸಮ್ ಕೋಡೆಡ್ ಲ್ಯಾಂಗ್ವೇಜ್ ಅಲ್ಲಿ ಒಂದು ಇನ್ಫಾರ್ಮೇಶನ್ ಸ್ಟೋರ್ ಆಗಿದೆ, ಅಂಡ್ ವಾಟ್ ಕೈಂಡ್ ಆಫ್ ಇನ್ಫಾರ್ಮೇಶನ್ is stored and in what form it is stored, that also we will now understand in this particular chapter. Right? Not only that, and how that information is being processed at various different steps, right? Like transcription andrenu, translation andrenu, and how during the cell cycle the DNA is replicated, a copy of the DNA is being produced during the replication, during the cell cycle, right? During cell division. So how the replication takes place, what is the mechanism behind it, what are the various enzymes involved in all these processes such as the replication, what is the transcription, translation, all these processes we are going to learn in detail in this particular chapter. right? So you know DNA and RNA. So when we talk about the genetic material, see, when we say the inheritance, you already know it is nothing but it is the process of transferring the characteristics from the parent generation to the offsprings, right? So for this, what is required? The genetic material is required, and that genetic material can be either DNA or RNA. Yes, either DNA or RNA. You have learnt in the chapter of biomolecules. DNA and RNA are what kind of molecules? They are nucleic acids. They are the nucleic acids. Yes. So nucleic acids are of two different types. One is called as DNA and other is called as RNA. What is the full form of DNA? Deoxyribonucleic acid. Deoxyribonucleic acid. And RNA is what? Ribonucleic acid. RNA is ribonucleic acid. Okay, so these two different types of molecules, how do they different from each other, and what are they composed of? The composition of the DNA or the making of the DNA, let us first understand, and that will make us easy to understand the structure of the RNA also. See, DNA for that matter, that is called deoxyribonucleic acid. See, this is made up of three main components. Three main components or three main ingredients that makes the DNA molecule are, the first one is the sugar. What kind of sugar it is? It is the pentose sugar. Pentose sugar anta helidre, it is consisting of 5 carbon atoms. Pentose sugar means consisting of 5 carbon atoms, and basically in DNA that pentose sugar is called as deoxyribose sugar. Deoxyribose sugar. For RNA, in the case of RNA, the same pentose sugar will be called as ribose sugar. Ribose sugar. So what is the difference between this ribose sugar and deoxyribose sugar here? See, eradu kuda pentose sugar molecules hai. Okay, here also there are 5 carbon atoms, and here also there are 5 carbon atoms only, right? So what is the difference? What to call them as deoxyribose sugar here, and why it is called as ribose sugar here? See, let me try to draw the structure, molecular structure of that molecule. See, this is 1, 2, 3, 4 and 5. There are 5 carbon atoms here, right? And so deoxyribose sugar means, deoxy refers to what? Removal of oxygen, yes or no? One oxygen atom has been removed from this pentose sugar molecule, and that is why it is called as deoxyribose sugar. So one oxygen atom remove agide, yellinda remove agide? From which carbon atom? The carbon atom at which place has lost the oxygen atom? See here, H, OH, H, H, okay, H, OH, then CH, 2OH and H. Here, look at this one, the second carbon atom, first, second, third, fourth and fifth, right? So second carbon atom nalli one oxygen atom kadime ide. Right? So instead of OH, only H is there. Whereas in this case, what do we see here? In this case, the pentose sugar molecule, the 5 carbon atoms of the pentose sugar, so they will be carrying HOH, 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 H and CH2OH. So this is the structure here. Here oxygen atom is present, that is OH is present, and instead of OH at a second carbon atom, here what is there? Only H is there. That is one oxygen atom is less, and that is why it is called deoxyribose sugar, and here it is called as ribose sugar. So this is the first component, remember, right? And the next component is the, which one? The nitrogen base. Nitrogen bases. See, these nitrogen bases are present in both DNA and RNA, but they, of course, the nitrogen bases, they have been divided into 2 different types. purines and pyrimidines. Purines and pyrimidines. See, under purines, we consider two nitrogen bases, that is adenine and guanine. Adenine and guanine, or sometimes they are also called as adenine and guanine. Okay, and are under the pyrimidines, cytosine and thymine are considered. Yes or no? Similarly, under RNA, that means the nitrogen bases that make the DNA are adenine, guanine, cytosine and thymine, whereas the nitrogen bases that make the RNA molecule, of course the same purines and pyrimidines, only one difference is there. The thymine is replaced by another nitrogen base called as uracil. So instead of thymine na badalu yaudide, uracil ide in the case of RNA, right? The same, see, the purines and the pyrimidines. So here the purines present here are what? Same, adenine and guanine, and these are cytosine and uracil. Cytosine and uracil are the pyrimidines. Okay, so uracil is present only in RNA and thymine is present only in the case of the DNA. And these nitrogen bases, they are, one more thing you should know, the purines are always double ringed structures. The purines are always double ringed structures, whereas the pyrimidines are always single ringed structures. The molecular structure, when you look at the purines, they're always double ringed structures and pyrimidines are always single ringed structure. This can be a very important information that you need to remember for the further understanding in this chapter, right? And so here these purines and pyrimidines, they come together in the DNA molecule or maybe in the RNA molecule specifically, right? And to the pentose sugar, maybe the deoxyribose sugar or the ribose sugar, to one of the carbon atoms the nitrogen base has to be attached. Yes or no? So to which carbon atom the nitrogen base is being attached on the pentose sugar? See, that carbon atom is nothing but the first carbon atom. Okay, the nitrogen base is attached to the first carbon atom of the pentose sugar, and by which bond? By N-glycosidic bond. N-glycosidic bond. Nodi, ಏನಾಗುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ, see, this is the pentose sugar, and to this pentose sugar the nitrogen base is attached. The nitrogen base is attached. Okay, the nitrogen base is attached with N-glycosidic bond. See, I have taken a double ringed structure here. That means it can be either the adenine or guanine, because purines are always double ringed structures. Yes, so if you take a single ringed structure here, see, I'm going to consider a single ringed structure here. See, with the N-glycosidic bond, a single ringed structure has been attached to this with the N-glycosidic bond to the first carbon atom, right? So this can be what? This can be either cytosine or thymine, because it is DNA, right? So these are the two components. Then what is the third component that makes either DNA or RNA? What is the third component that is being present in the nucleic acids? The third component is nothing but the, which one? The phosphate group. The phosphate group. See, this phosphate group is actually a phosphoric acid which got ionized. So phosphate group is also called as the phosphate ion that comes and attaches to the either the fifth carbon atom or third carbon atom. Usually ನೀವು ತಲೆಯಲ್ಲಿ ಇಟ್ಕೊಳ್ಳಿ, fifth carbon atom ಅಟ್ಯಾಚ್ ಆಗುತ್ತೆ ಅಂತ ನೆನಪಿಟ್ಕೊಳ್ಳಿ. Okay, the phosphate group attaches to, this is common for both. Phosphate group attaches to the fifth and third carbon atom. So ಹಾಗಾದ್ರೆ fifth and third ಅಂತ ಹೇಳಿದ್ರೆ, are there two phosphate group attached to one sugar molecule? Yes, but only in a chain of many nucleotides. See, we are giving different names for them. I think you have already studied that in the biomolecules chapter. See, DNA, that is the pentose sugar, maybe the deoxyribose sugar or the ribose sugar, along with the nitrogen base will be called as nucleoside. Okay, I am going to write that information here. See, the pentose sugar plus nitrogen base will be called as nucleoside. It is called as nucleoside, whereas to that nucleoside, when the phosphate group comes and attaches to the fifth carbon atom, then it will be called as what? The nucleotide. See, nucleoside plus phosphate group will be called as what? A nucleotide. So phosphate group ಹೇಗೆ attach ಆಗುತ್ತೆ? See here, the fifth carbon atom is here, right? So the phosphate group attached to the fifth carbon atom, right? With which bond? The bond here will be called as phosphodiester bond. The bond here will be called as phosphodiester bond, right? Phosphodiester bond anta heltaralla, DNA has got the phosphodiester bond to hold the phosphate group anta ನೀವು ಓದಿರಬಹುದು ಅಲ್ವಾ? See, phosphodiester bond yavaga anta helidre, see, this is one nucleotide now. See, this has got the nitrogen base attached to the first carbon atom with the N-glycosidic bond, and this has got the fifth carbon atom attached with the phosphate group with the phosphodiester bond, right? Now this is one complete nucleotide. So when many nucleotides come together, ಒಂದರಿಂದ ಮತ್ತೊಂದು nucleotides ಬಂದಾಗ, all these nucleotides join together with the help of one common phosphate group. So ಎರಡು ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಗಳ ಮಧ್ಯದಲ್ಲಿ one common phosphate group will come and it forms a chain of nucleotides, and that chain of nucleotide is nothing but, or it is also called as a polynucleotide chain ಅಂತ ಏನು ಕರೀತೇವೆ, that is what is nothing but a nucleic acid. Okay, if that nucleotide is consisting of the deoxyribose sugar, then the nucleic acid can be called as DNA. If that polynucleotide is consisting of the ribose sugar and uracil, then it can be called as RNA. Yes, so alli enagutte, see here, when this is one nucleotide, some nitrogen base is there, right? I'm going to write N there, and there is one phosphate group. There is one more here. See, there is one more here, and this also has got the nitrogen base, right? And this also has got the phosphate group at the fifth carbon atom. I'm going to write one more here. See, this is also one phosphate pentose sugar with nitrogen base, right? And the phosphate group at the fifth carbon atom, right? So here phosphate group, ಎಲ್ಲ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಗಳಲ್ಲಿ phosphate group is attached with the help of which bond? The phosphodiester bond, and the same phosphate group will go and bind with the next successive nucleotide to the third carbon atom. Illi the third carbon atom is having the free OH group, right? So in this case also the third carbon atom will provide the free OH group, so which provides a site for esterification or bond formation. Which bond? Phosphodiester bond. That is, phosphodiester bond formation takes place by esterification. So E phosphate group ಏನಾಗಿರುತ್ತೆ, this is the phosphate group present at the fifth carbon atom of this particular nucleotide, and the same phosphate group will bind with the, or it is going to form bond with the third carbon atom of the next nucleotide, and this one also forms with the next nucleotide. Now there are two bonds, phosphoester bonds. So that is why in a chain of nucleotides, in a polynucleotide chain of DNA or RNA, the bond present will be called as phosphodiester bond. The bond now it can be called as what? Phosphodiester bond. The phosphodiester bond formation always takes place. Phosphodiester bond always takes place in one direction, that is 5' to 3' direction. Idu hege anta nanu matte heltini nimage. So for now, just keep that in mind. Phosphodiester bond formation takes place in this direction, that is 5' to 3'. Okay, so this is the basic information that you need to know about the genetic material. So you just understood how the DNA or RNA is made up of. That means the different components are there. So what are the three different components that we just studied? That is nothing but, that is the pentose sugar is one thing. Pentose sugar can be either deoxyribose sugar or ribose sugar. Deoxyribose sugar or ribose sugar, right? So it depends which type of molecule it is, and based on that it will be consisting of either the deoxyribose sugar or ribose sugar. You know, it is in DNA and it is in RNA, and along with that the nitrogen bases will be there. So there are 4 nitrogen bases, right? So these 4 nitrogen bases, they are divided into purines and pyrimidines, right? So they are attached to the first carbon atom, attached to the first carbon atom of the pentose sugar, right? With which bond? With N-glycosidic bond. With N-glycosidic bond, right? And the third component of any nucleic acid is what? The third component is nothing but the phosphate group. The phosphate group that is attached to the fifth carbon atom with which bond? With phosphoester bond. With phosphoester bond, right? So niಮಗೆ ಗೊತ್ತಾಯ್ತು. See, this is, I'm going to draw one representational thing now. See, this is the pentose sugar now. To this pentose sugar, the first carbon atom is being attached with a nitrogen base. Okay, a nitrogen base. Okay, maybe adenine anta tagolona because it is a double ringed structure. The bond that is present between the first carbon atom and the nitrogen base is which bond? It is the N-glycosidic bond. Remember, these can be very important 1-mark questions and also important CET and NEET questions. Yav bond ellirutte? So for the attachment of which particular molecule, which bond will help? That is very important for you to remember, right? And here to the fifth carbon atom, here the attachment of the phosphate group can be seen, and the phosphate group will the attachment of the phosphate group is helped by the phosphoester bond. Phosphoester bond, right? Phosphoester bond. So when this is going to form a nucleotide chain or a polynucleotide chain anta ಏನು ಕರೀತೇವೆ. See, just see now, I'm going to draw a polynucleotide chain here. So let me consider many pentose sugar molecules. Okay, so all of them, they have got what? Nitrogen bases, one or the other type of nitrogen bases, maybe the double ringed or single ringed nitrogen bases. Okay, and they have got the phosphate group attached to the fifth carbon atom. They have got phosphate group attached at the fifth carbon atom, that is of each what nitrogen base, sorry, each pentose sugar molecule, and here this phosphate group is going to form one more phosphodiester bond with the next or successive nucleotide. This is one nucleotide alva, this is one nucleotide, yes or no? So this is one nucleotide. So when there are many such nucleotides are formed, so all of them are joined together with the help of phosphodiester bond, right? Phosphodiester bond, right? So this is nothing but a polynucleotide chain. So polynucleotide. So in this polynucleotide, what you are observing here, see the phosphate group, E phosphate group and observe madidre, this phosphate group is attached to the third carbon atom of this particular pentose sugar and to the fifth carbon atom of this particular pentose sugar. So you also observe this one, this is also completely attached on both sides, this is also attached on both sides, but look at this one. So this phosphate group is free because that is the last nucleotide, and that is why it is free at the fifth carbon atom, and that is why this is called as 5' end anta kariteve, 5' end, or it is also called as 5' phosphate end anta kuda kariteve, 5' phosphate. And similarly you see the other end, what is the other end here? What will be there? Third carbon atom irutte alva. Third carbon atom will be free from. Third carbon atom, it is also free. Sorry, not this one, this one. The third carbon atom, it is not attached with the phosphate group, and that is why this is 3' OH group anta kariteve, athwa 3' hydroxyl group anta kariteve. Yes, so this is how the formation of a polynucleotide chain is formed. Okay, the polynucleotide chain is done in this way, right? So it can be in DNA or it can be in RNA. The only difference I have already told you with the structural differences when we talk about the DNA and RNA. One difference is what? The pentose sugar is different. The other difference is the thymine is present in DNA and uracil is present in the RNA. Okay. In the next session, I will talk about the DNA double helix model, who proposed it, all that let us consider in the next class.